Welcome to my exam mini video 2 on epistasis. What is epistasis? First, we need to cover key vocabulary. First, we have gene. It's inherited factors from our DNA that determines our characteristics. It's basically like instructions for our body. Next, we have loci. It's places on chromosomes where different forms of the genes are held. As you can see from this picture, the highlighted area are those places where the genes are located. Next, we have phenotype. It's the physical trait expressed from the gene and genotype. It's what you see, like blue eyes. Next, we have genotype. It's the sets of genes for a certain trait, like 2 big T's are tall, 2 little t's are short. Now, back to epistasis. Epistasis is a type of gene interaction where 2 genes at 2 loci contribute to the same one phenotype by one gene hiding or masking the other gene at the other loci. Basically, it's where multiple genotypes lead to the same phenotype. But how? So for some traits or phenotypes, such as having straight hair, one genotype from a specific gene produces its own polypeptides to represent one phenotype, versus like to the right, where traits like hair color can have 3 different genotypes with 3 different polypeptides and still represent one phenotype too. Now that we have a better idea of epistasis, we are now going to go over one of the 6 modes. Using the story of the Little Mermaid, as we all know, Ariel has 6 other sisters, and as you can tell from my drawing, they each have different hair colors, some with blonde, others with dark, and of course Ariel with red hair. How can 7 sisters who will have the same 2 parents have 3 different hair colors? This is all because of epistasis, and specifically single dominant epistasis. That is the mode that we'll be focusing on today. So the single dominant pathway looks like this. It has 3 possible trait outcomes for our phenotype of hair color based upon their correlating genes found on their loci. Each genotype, as seen here on the left, goes to the pathway to find its correlating genes that will lead to the phenotype based on if it is inhibited or not. So single dominant has a 12 to 3 to 1 ratio that results from a dihybrid cross. Also, we had the breakdown of the genotypes to the left with the correlating hair color, blonde, dark, and red. Now we're going to explain the proportions of 4 genotypes, the ones you see at the top of the page here, that come from crossing our hair color genotypes based upon the mode of single dominant epistasis. And the first cross of heterozygous dark hair with heterozygous dark hair, we get a 9 to 3 to 3 to 1 ratio. And the second cross between the heterozygous dark hair and blonde hair, we get an 8 to 0 to 8 to 0 ratio. So if we simplify our ratios, we get a 12 to 3 to 1 ratio at the top cross, meaning that it is single dominant epistasis. But we get a 1 to 1 ratio for the second cross, meaning it's not single dominant. Thank you so much for watching.